Hi, in this video, we would look at two-hit hypothesis which is related to cancer progression. So this hypothesis was given by Alfred Knudson, who was an American physician and a geneticist. So this hypothesis is stated as most genes require two mutations to cause a phenotypic change, right? We would understand what that means. Before that, we try to understand what was his observation. So as a physician, he observed few things. So he performed statistical analysis in case of retinoblastoma, which is a tumor generally present in the very young child, and it could be inherited or it could be sporadic in nature as well. So this is how a normal eye look like. In case of retinoblastoma, the retina looks like this. So it's a cancer of eye. He found that retinoblastoma, which could be both familial and sporadic, but in case of the familial retinoblastoma, people who had history of retinoblastoma in their family, the inherited retinoblastoma occurs at way younger age than the sporadic cases. So the individuals are getting affected by this tumor at a very early age. But in case of sporadic, the onset is late. In addition to that, the children's which have like inherited format of retinoblastoma or a familial form of retinoblastoma, in that case, the phenotype is severe. Both the eyes are affected. Based on these two hypotheses, he thought that there might be some predisposition in case of the familial retinoblastoma, which is augmented by other reasons. Whereas in case of sporadic situation, there is no predisposition. That's why the augmentation is a bit more difficult, and that's why it is mostly occurring on the adult stages, not in the very young stages. So now we know that any of the gene has two versions, right, in two different chromosomes. So the gene PRB also has two versions. Now both these cases, one of the possibilities, like one of these versions is mutated, right? So the protein that is produced from the version would be not produced, but there is another version of that gene which is functional. So some amount of protein production would be there. Now let's say there is a problem in both the versions. As a result, either the production won't be there or a mutated form of protein would be produced, right? And as there could be a different consequences in terms of phenotypes. In case of familial retinoblastoma, where the retinoblastoma onset is very early age, there what happens is there are like already one genetic mutation in the PRB gene. The probability that the second copy gets mutated is very high, and that's why mostly these retinoblastomas are seen in early ages. Whereas in sporadic retinoblastoma, both the copies are initially fine. Now, in order to get two simultaneous hit in two different locus, the probability is pretty low. So it takes a lot of time. So generally, the sporadic cases of retinoblastoma occurs normally at like adult ages, not generally in the young ages. Okay, so we learned about the bit about the genetics behind the two-hit hypothesis. But what is the cell biology behind two-hit hypothesis? What actually is performed by retinoblastoma such that it leads to a cancer? So we know that the initial phase of the cell cycle is the interphase, and in the interphase, the longest phase is the G1. In G1 phase, the cell receives signal from the environment, such as from growth factors, and decide whether it has to divide or not. If the growth factor is present and the environment is permissible for division, then what happens is the cells would first transcribe cyclin D. Now the cyclin D would phosphorylate a tumor suppressor gene, which is the PRB in our case. Once PRB is suppressed and phosphorylated, then E2F can lead to transcription of S phase cycling, which would allow for progression from a G1 to S. Now imagine we have this normal PRB gene and normal PRB gene product which is inhibiting E2F. So it would inhibit growth. It works like a tumor suppressor, right? And the way it inhibits growth is not allowing the cell cycle to progress from G1 to S unless and until there is a growth factor in the environment, and in presence of growth factor this inhibition is withdrawn. But think about a situation where this PRB is mutated. In that situation there is no one to prevent E2F from binding, so it doesn't require any growth factor signaling for PRB mediated suppression to go away. It looks like the E2F is constitutively active, so the cells, no matter how they receive the signal from the outside, they always decide to transit from G1 to S, and as a result uncontrolled growth takes place, which is very much similar to a phosphorylated format of PRB, where PRB cannot inhibit E2F and E2F is free to transcribe cyclines which are required for S phase, and as a result G1 to S transition happens. And as a consequence of all of these, the tumor forms in the retina. And that's the story behind retinoblastoma and two-hit hypothesis. I hope this video was informative. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.